percentage of fish that don't make it all the way to spawning has been anywhere between 30% to 95% in different stocks. And that varies a lot in different years, and there's really not a high degree of predictability of what years are going to suffer those very high levels of mortality. There appears to be some relationship with temperature and high-level temperature stress in the river, which has also been on the rise, but we don't have a really clear understanding or predictability for which years will have the highest levels of mortality. So we undertook this study to ask the question, could we predict in advance of fish entering the river, or at the point that the fish are entering the lower river, can we predict which individuals are going to make it to spawn? So are those fish physiologically challenged before they enter the river, or is what is undermining their performance in the river something that they're coming upon after they enter the river? So we tagged fish in two environments in the marine approaches to the Fraser River, and we tagged them also in the lower Fraser River, and we tagged them at the spawning grounds. And then we had receivers throughout the Fraser River that are marked in red, and we could track their speed of migration, when they entered the river, how far they traveled, and when they disappeared in the river. We conducted genetic stock ID on all of the fish that were tagged, so we knew exactly what stocks they were going to. We used microarrays to look at the difference between fish that made it to the spawning grounds versus those that went missing en route to the spawning grounds. And interestingly enough, we found in these three independent studies for the tagged in three different environments that the same genomic signature was associated with premature mortality in the Fraser River. And this signature was most predictive of mortality in fish before they entered the river. So this was key, because that's ideally what managers want to know. Is we want a tool that will allow us to predict mortality well before the major fishing impacts occur. So this particular signature shows some level of variation in different stocks, but was fairly profound in quite a large scale number of fish. So a functional analysis of the signature revealed that the most plausible explanation for the genes that were being stimulated was response to a viral infection. So we have looked at other tissues and other years in other experiments, and we have identified that this same signature that we identified in the gills of these migrating sockeye salmon have also been identified in other tissues, in brain tissue, in liver tissue, and in gill tissue, but not in muscle tissue. And interestingly, it's also identified in multiple life history stages of the salmon. And in large-scale studies in smolt, brain, and liver that were conducted in 2008, we identified this signature and found that the prevalence of this signature it decreases over migration during the first 3 months in the ocean. So the smolt showed signs of the MRS signature well before they left the river, and even as par, about 2 months before they were smoltifying. And the highest prevalence were seen at the time that they were leaving the river, with a significant drop in prevalence in the fall in the ocean. There was a 30% reduction from summer to fall in the brain, and a 50% reduction in the liver. Now there's two possibilities to account for this reduction. One is that the fish simply recover from this signature, and another is that there could be mortality related with fish that were positive with this signature. This is something that we're ongoing looking at in more fish. We hypothesized that this signature was caused by a virus, and so we took tissues that were positive for this signature, and we did high-throughput 454 sequencing on those tissues. And we identified a novel salmon parvovirus. Parvoviruses have never been observed in fish before. That was found in this MRS-positive tissue. We have about half the genome sequence of this parvovirus, and we're continuing to do more research on this. We repeated our tagging study in 2010, and we have looked at the prevalence of this parvovirus in that tagging study and found that 30% of the adult gills were parvovirus positive before entering the Fraser River. And importantly, one of the stocks, the early shoe top stock, which is the stock that was most adversely affected by the MRS as well, at a 9.5 times lower probability of arriving to the spawning grounds if they were positive for this parvovirus. So the parvovirus was a differential to explain premature mortality of this stock. Now, another observation is in the smolts of out-migrating smolts with the parvovirus, and we do see the parvovirus also in the same tissues that we observe the MRS, in the brain tissue, the liver tissue, gill tissue, but also in kidney tissue and heart tissue. And interestingly, there's only one stock in the Fraser River that's increasing in productivity, and that's the Harrison River stock. And this is the one stock that we have been unable, in multiple years of study, to find any evidence of the parvovirus in the smolts that are leaving the river. Now, given that we believe that a lot of the mortality or the variable mortality that's hard to predict is occurring in the early marine environment, and this is the only stock of fish that don't carry this virus in that phase of their life history, this is again another differential to consider. And we're continuing research on this parvovirus. Now, something that came out in the Cohen inquiry quite clearly is that we really don't have a good feeling for what pathogens wild salmon are affected by. And the main reason for that is that we don't see wild salmon die. Now, we know a lot about disease in aquaculture fish because they're raised in hatcheries and then they're raised in net pens, and we actually visualize those fish dying. And so most of what we know about disease comes from what we know about aquaculture. There's some information about hatchery fish in coho and Chinook salmon. If there are large mortality events in freshwater, we sometimes understand or do studies on the diseases that affect them. But again, even aquaculture fish, as soon as they move into the ocean, we don't see them die, and we know very little about what pathogens may negatively affect them. And so after finding this novel parvovirus, and we have a number of other genomic signatures that indicate differential immune stimulation, and we're interested in knowing which of those may be involved in pathogen response. We set out to use genomics and use a high-throughput genomic screening tool to look at all of the pathogens that we know about for salmon throughout the world, and whether or not which ones of those may be affecting our salmon here. So we have a Fluidigm biomark system which allows us to look at 96 different individuals and 96 different assays at once. And we went to the literature and found TaqMan assays for a wide variety of viruses, bacterial pathogens, and some small parasites. And we began employing these with the Fluidigm biomark. In addition, at the same time, we have a number of biomarkers that are predictive of our MRS signature, which we are tracking now over thousands of fish. And so we parred the pathogen screening and the MRS signature into the same runs. And what you can see here in the ones to the left are these are the various pathogens, and they're run in duplicate, MRS to the right, and I've starred the lane that shows the two replicates of the parvovirus. And the yellower that you see, the higher copy number you have for the pathogen or the biomarker. And you can see that these are actually smolts leaving the Fraser River, and including some sample in the marine environment in liver tissue. And you can see that these smolts carry a lot of pathogens. And all of the pathogens used in this particular screening were pathogens that are known to cause mortality in some environments. Okay, so they weren't your avirulent pathogens. And you can see that in the liver tissue, the parvovirus is one of the most common pathogens that were potential pathogens we've observed. We still are doing the challenge work to determine pathogenicity and infectivity of this. But there's a number of others, and I have in a previous slide, I didn't do it here, but there's about 4 of these that you're seeing here that we didn't know exist in BC. One of them is highlighted under the industry samples here, and that is a virus that has been observed in Norway, and we did not know was here in BC. And you can see it has a fairly high copy number in some of the industry samples that we've looked at, but it's also present in wild fish. We've done this over over hundreds of samples in gill tissue and in liver tissue, and we're moving on to brain tissue, and we're working on adults and smolts to try to understand. The question is, what pathogens are these fish carrying, and at what kinds of copy numbers, so what kinds of intensity of infection. And so one of the things that I'm interested in is contrasting the pathogens present in different years or as different differentials. And so 2007 again pulls out as the year where we had very, very low returns, and I have starred the pathogens that were present in liver tissue, or sorry, in gill tissue, that were a differential for 2007 compared to other years. One of them is a Flavobacter, a bacterium that's quite pathogenic in the freshwater environment. And another is a PRV as a virus that stands out in 2007 in the marine environment, but isn't present in the freshwater environment. These are the kinds of things that we're moving towards in terms of trying to build a better understanding of what pathogens may be negatively impacting our wild fish. This is really a discovery phase. This isn't doing the ultimate challenge work, but it's a way to start to rule in what pathogens we should be paying attention to in wild fish. There's a second part of this that is made only really available because of the genomics program, because all of these fish that we're screening, or many of them, we've already run on microarrays. And so now we're also employing retrospective genomic analysis to ask, to again try to par down the pathogens to ones that might be the most important by looking at the degree of host response to those pathogens. And so we're looking at genes that are associated with copy numbers of various pathogens. Here I show an analysis that was done by a postdoc in my lab, Brad Davis, on Nucleospora, and finding a genomic signature in wild fish associated with Nucleospora copy number. And so this is going to be a very rapid way to begin to understand how these wild salmon are responding to a large variety of pathogens that we can screen very quickly. And we're using this data to rank the potential impacts of pathogens and which ones we should carry through with further challenge studies. Infectious salmon anemia, anyone who's followed the Cohen inquiry in the last couple of months, this has been a real issue in wild salmon, the discovery of this infectious salmon anemia virus, and it's been hotly debated whether it really is here or not. My lab is one of the labs that had evidence that this virus actually is here in BC, or something like this virus is here in BC. And we have sequence confirmation of a couple of the segments, short reads of a couple of the segments, but we've done a lot of PCR screening now for this. And again, we find that the ISA7, segment 7 primers, show a high prevalence of this particular virus in 2007 relative to some of the other samples. And if we run a retrospective genomics analysis, we see that we have a very strong genomic signature to this. And if we do a functional analysis of that genomic signature, the very top hit is influenza infection, and ISA is an orthomyxovirus, which is an influenza virus. So basically, these salmon are responding to something that resembles the flu in mammals.